So, welcome everyone to this INNS webinar. Today will be a great presentation about actually our nervous system. I'm very nervous about that. Jonathan Wolpaff will speak about it, but before introducing to the talk, just generally, this is hosted by the International Neural Network Society. You can find us under the barcode there or just typing inns.org in the browser. And there you will see not only this information of that talk here, but many other events and a link to that YouTube channel here where you can follow up of the presentations. Also, that one here is being recorded and you can share it afterwards if you think that was quite interesting. I'm pretty sure it is, because we'll hear about Heksor. But thinking a bit by myself when I was learning, for example, cycling, right? It takes a while, but at some point, we kind of got it. And then when you have a new bicycle, it goes rather quickly to adapt. And over time, even though you might not bicycle for a few years, you still have it somehow inside. And how this is happening, the answer has been found already a few years ago. This idea of Heksor. I will not go into the details. We have the expert with us in a moment. But the main idea is that there is a system or like a network inside of our nervous systems which allows to store these locomotion plans. And one of the key principles behind is that plasticity of this Heksor ensures actually a stability of the memory. Like if changes are there, the core plan is not, it stays stable. But how that really works and how these key features are maintained while others are being constantly adapted and negotiated, that's still unknown. And there's this recent trend to look into using neural networks to further exploring that. And who could speak better about that than the key investigator in this research himself, Jonathan, who is with us. He will share some of his thoughts, but he has actually already worked in this area. That's amazing. That's longer than I am old. Like since 1969, he is interested in memory, and he has done more than 50 years of research. Now he is in the University of Albany in New York, and his experience is really long-standing, and he has in pre-discussions with me pointed out research papers which kind of have been totally forgotten already 40, 50 years ago, which are one of the key ideas behind of this algorithm, or this whole theory. An interesting thought or outcome, what he is very proud of, is that this paradigm is generating a new framework for neurohabilitation, which is maybe now already enhancing recovery for people with spinal cord injury, stroke, and other chronic neuromuscular disorders. So if you have not been interested so far, I think that is extremely interesting as well. I'm very curious to hear more about it. Please, Jonathan, the floor is yours.
Okay, well, thank you very much, Marcus. I'm really very pleased to be here and have the opportunity to talk. Now, I know I'm talking here to people who have different perspective, a different background from mine, which is not normally the situation when I give a talk. So it's very exciting because I'm really interested in your reaction to this. I'm interested in perhaps engaging you to participate in what is really a very exciting endeavor and certainly needs input from multiple disciplines to go forward. I sort of feel like I've been in this seminar for a while because I've been going back and forth with Steve Grossberg about various aspects of my talk. So I'm really, I think, I'm really into the situation. But now I'd want to share my screen. And I think I'll get it right. I don't think that's right.
No, it's not yet. We see the upper half of the
Yeah.
PowerPoint view itself.
So how do I unshare? It was working a few minutes ago.
Yes, I think you just go to the Zoom menu and make a new share. Click on the share button again and select the.
Um,
Keller, you can pause the recording for a moment.
Thank you.
Perfect. Then go ahead.
Okay, so the title is Heksor, CNS substrate of a skill. Plasticity ensures stability. So, as we presently understand it, the function of the central nervous system is to acquire and maintain skill behaviors or skills. These range from simple skills such as the flexion withdrawal reflexes that are acquired in utero and early postnatal life, through standard skills such as locomotion and speech, to the most sophisticated athletic, artistic, and intellectual performances. This is what, by our understanding, is the nervous system does. So we have all these billions of neurons, and their job is to produce appropriate outputs from a few 100,000 spinal motor neurons. I'm ignoring for a moment hormones, which central nervous system also puts out, but these several 100,000 spinal motor neurons, their activity is the result of what all these billions of other neurons are doing. That's the output, that's the function. So how are skills acquired and maintained in the central nervous system? So 50 years ago, the problem was straightforward, and people who weren't around 50 years ago really can't understand how much neuroscience has changed. It's really like a revolution over the past 50 years. The picture has changed completely. 50 years ago, it was generally accepted that the CNS is mostly hardware and that skills are simply programs that are stored in these very special modifiable synapses that are in special parts of the brain. And these programs are accessed when they're needed. They're just downloaded when they're needed. Thus, the problem is to find and to characterize the modifiable synapses. That was not a solved problem, but it seemed like a very straightforward problem. Now the problem is considerably more difficult. First, we know that the central nervous system changes continually throughout life. Wait just a second. Yeah, there's neurogenesis. There's gene activation, neuronal properties change, synaptic properties change, there's long-term potentiation and depression, glia change, dendrites change, hormones change, there's sprouting that occurs of axons and even vascular changes. Now, all these changes somehow get coordinated and synchronized to produce and maintain all our skills. How does this happen? A second thing is that it's become clear that a skill is not exactly the same way every time. This is Nikolai Bernstein, whom I think many of you know. He was a very famous Russian physiologist in the 20th century. And he showed that skills, among many other things, that skills are not exactly the same every time. Here you see a skilled person swinging a hammer to hit a chisel at two different places, and the trajectory varies. But what doesn't vary is where the where the hammer hits. The hammer hits every time on the chisel. The rest of this trajectory varies. It really doesn't matter. The important part are exactly where it hits. So the trajectory varies, but what doesn't change is where the hammer hits. This is the key feature of the trajectory. This is all that really matters. Exactly where the hammer hits. This defines the trajectory as satisfactory. So it became clear that skills have key features that define them as satisfactory. They aren't exactly the same every time, but they maintain these key features if they're to be satisfactory. So there's been much attention and substantive progress on these two new aspects of skill acquisition and maintenance. There have been physiological models for specific skills, such as perhaps one of the best known is for locomotion by Grillner et al. There's been studies of ongoing adaptations during performance, how they're continually adapting to keep so that the key features are maintained. John Krakauer and many others have done many studies of this. They're neural network models, as people here know very well, for a wide variety of skills. Steve Grossberg has been one of the primary leaders in this. So at this point, so I've gone through those two major changes in the past 50 years. At this point, I want to take a little digression just to introduce the simplest skill behavior, which I'll be talking about and using the implications of a great deal from then on. Really, the simplest behavior of the mammalian central nervous system is the knee-jerk reflex. This is a reflex that's produced by stretching the muscle, or it can be produced electrically by simply stimulating the afferent nerve. This is called the H reflex, after Hoffman, who described it now something over 100 years ago. The afferent stimulus goes in, it synapses on the motor neuron, largely monosynaptically. It produces the H reflex. If you do it electrically, it's the stretch reflex if you do it by stretching the muscle. Now, this is the M wave, which is basically just from stimulating the largest efferents. So that precedes it. This is in a rat. This is the gastroc muscle in a rat, or actually the soleus muscle in a rat. So it's occurring about 6 to 10 milliseconds after the stimulus is given. It goes in here and it produces the reflex. It's totally spinal. It's the simplest possible behavior. At the same time, it's under control of descending control pathways from the brain, so that this reflex can be affected by this descending control. And what we and others have done, starting actually in the 1980s, is shown that monkeys, rats, mice, and people can gradually change the stretch reflex size or the H reflex size when they're rewarded for doing so. This was originally shown in the monkey stretch reflex in the early 80s. Steve Wolf and Rick Siegel showed it in the human stretch reflex a little later. The H reflex was then developed in 1987. But the phenomenon is pretty much the same in all these species, both for the stretch reflex and the H reflex. So this is H reflex conditioning in our laboratory by Shenyan Chen, showing these are many rats in which we reward them for a larger H reflex over days or a smaller one, and the size of the reflex changes. And this is in people, the same thing, the H reflex getting bigger over days, getting smaller over days with down conditioning, and preservation of this in follow-up several months later. This is from Iko Thompson's work. So now that I've introduced that simplest behavior, I'm going to introduce the third big change in the last 50 years, which is really the core that's led to the presentation I'm making today. We spent a lot of time, Shenyan Chen and others in our lab, spent a lot of time looking at the plasticity that occurs when we make this simple change in the reflex. We make it bigger or smaller. And what we're doing is we're making a simple change in this reflex pathway here. And the pink areas show all the places of plasticity, all the things that change when we change this little reflex. There are changes in the cerebellum, there are changes in the sensory motor cortex, which changes cortical spinal tract activity, which produces a whole panoply of changes in synapses, as well as in the motor neuron itself. The spinal motor neuron changes, its threshold changes, there are even muscular changes that occur. So this is a very complicated network of plasticity that underlies this. Similarly, this is another simple skill from Julian Dion's lab, finger flexion sequence learning. This is people learning to flex their fingers in a certain sequence. As they acquire this skill and get better and better, the colored areas show the places of the plasticity that correlates with this increase in skill. So there are changes in cortex, there are changes in the basal ganglia, there are changes in cerebellum, and there are changes in the spinal cord. This is, again, is a distributed pattern of plasticity that underlies this skill. And the thing this slide is supposed to points out is that these two skills overlap. Their networks overlap, and they certainly overlap in the spinal cord. They're affecting the same neurons and synapses. They share the same neurons and synapses. Skills share CNS neurons and synapses. That means that when one skill changes, the others are affected. Thus, they too must change. Now, I can show this most graphically, really, in recent publication from our lab in work led by Yu Wang, looking at the axon initial segment of the spinal motor neuron. This is the most shared structure in the central nervous system. This is what determines whether the neuron fires, when and whether it fires. This is what starts the axon potential in the motor neuron. So this is the final gatekeeper for action. And here we see the axon initial segment. Here's the spinal motor neuron. Anchorin G is really a marker for the sodium channels, which are what determine when the sensitivity, the excitability of the axon initial segment. So if that's higher, that indicates that the axon initial segment is more excitable. The cell is more likely to fire. This is PP38, another protein that produces internalization of anchorin G in the sodium channel. So as it gets higher, the excitability goes down. There are GABAergic terminals on the axon initial segment. So this is normal. And here we see it going out, and here it's producing an H reflex. If we train this rat to make the reflex bigger, the reflex gets bigger, and there are changes that occur in the axon initial segment. And there's the two main changes that she identified, the significant changes are the axon initial segment gets longer and it gets farther from the soma. Now, there have been a number of groups that made modeling studies looking at the supposed effects of this. The upshot is that these effects should make the motor neuron more excitable and would therefore account for the larger H reflex. So this is up conditioning. Down conditioning is completely different. Here's the reflex that's smaller. What happens is anchorin G has gone down, PP38 has gone up, GABA input to the axon initial segment has gone up. All these changes make the segment less excitable. There's also increased GABA input to the motor neuron itself. So this appears to account, at least in large part, for why the reflex is smaller. The other thing that's interesting to notice here is that the reflex is a little later. That's because the change affects every node of Ranvier as it goes out. So that it takes a little longer at each node of Ranvier to depolarize. So therefore, it's a little slower. And this accounts for the reflex being a little slower. We modeled this several decades ago with John Halter at Baylor, and he had a model which really predicted this pretty exactly, that a change in the firing threshold would produce the given change in conduction velocity. So here we've done this really very simple little conditioning. We've changed this reflex, and we've changed the final gatekeeper. So every skill that uses this motor neuron has got to adjust. It's going to be affected by this simple little training that we did. So we have new knowledge that the central nervous system is plastic through life. The details of skills vary. The key features are what's maintained. The substrate of a skill is a network of neurons and synapses. Now, these networks overlap. They share their neurons and synapses. Thus, a change in one requires changes in others. This raises a new question. How do they all manage to maintain their skills? This really demands a new paradigm. And I think the paradigm is one in which these networks have two special properties. There's an individual property and there's a collective property. So the individual property is that the network changes continually to maintain the key features of its skill. So for locomotion, key features are things like symmetry, posture, balance, metabolic cost, etc. The EMG and kinematics may change, but the key features are maintained. These key features are the memory of the skill. The plasticity of the network ensures the stability of the memory. And then there's a collective property. All the networks are changing concurrently. They're changing the networks and synapses. They all share. Thus, the aggregate process is a negotiation. The networks negotiate the properties of neurons and synapses they all use. They keep the central nervous system in a negotiated equilibrium that maintains their skills. This is not really an equilibrium. This has been pointed out to me by my wife, who's a chemist. That's really a steady state. But it's similar. This negotiated equilibrium is similar to a Nash equilibrium as defined in game theory. It means every skill or every player really in this game is in the best situation that it can be. If it makes any change, somebody else is going to change and it won't be in that best situation. So this is the best situation for keeps all of them reasonably happy. It's a good enough solution as Jerry Loeb would describe it. So Heksor plasticity ensures a stable or a satisfactory negotiated equilibrium. So the CNS substrate of a skill is a distributed network of neurons and synapses that produces a skill and changes continually to maintain it. This was presaged by Nikolai Bernstein in 1967 when he said behaviors are biodynamic structures that live and develop. Together, these networks maintain a negotiated equilibrium in the CNS. It became clear that these networks needed a name that implies their special properties. And I was fortunate enough to find Adam Camisar, Hebrew Union College, who was a classical scholar who got interested in this problem. And it was an interesting interaction. It went on over months during the pandemic. We've never actually met. I hope we do in person at some point. But after considerable research, he identified an ancient Greek word, hexis, which was a term that was originally defined by Aristotle and then modified by the Stoics in the subsequent couple of centuries to indicate the concept of a substrate that was really the fine matter of the brain that was the substrate of a skill. And they thought about this as playing the flute or riding horses or whatever. In any case, it was a skill and it could change. It could get better. So it was something that was plastic. It was the substrate of a skill. It was in what they would call, or we would now interpret in our terms, as the fine matter of the brain. So they had defined this term 2,200 years ago. These people have been dust for 2,200 years, but it didn't matter because basically Adam was able to elide the 22 centuries so that these people could have been colleagues that we met in neuroscience last year. And we learned the term then and we subsequently used it. So that was a really kind of neat thing about this whole enterprise. In any case, from the word hexis, we're calling them Heksors. And the KS is basically equivalent to X in Greek. And that moves it away from things like hexane or hexagons. And the OR means it's an actor. It's an active agent, like an actor, an effector. So a Heksor is a distributed network of neurons and synapses that produces a skill and changes continually to maintain it. These are Heksors. This is the Heksor to the extent of our knowledge of them now. For finger flexion sequence learning, this is for spinal reflex conditioning. So what is a Heksor not? A Heksor is not a memory or an engram. Memories are passive. Heksors are active. In 1975, the idea was the central nervous system uses memories to produce behaviors. Now, we understand that the central nervous system is essentially multi-user equipment. That's what it is, and its users are Heksors. The other way to say it is that the central nervous system is an environment, it's a venue, and the inhabitants of this environment are Heksors. The key features of a Heksor skill are a memory. Heksor plasticity ensures memory stability. A Heksor is not an alternative to other theories about specific skills. The Heksor negotiated equilibrium addresses a different problem. It's simply the problem of how do skills survive in the continually changing central nervous system that they share. And the paradigm doesn't really specify the accommodation among skills. The paradigm proposes the process that reaches the accommodation. So this is a empty nervous system. It has multiple places, all those deltas that can change, synapses and neurons. Here's one Heksor early in life. We acquire flexion withdrawal reflexes that withdraw your foot from a thumbtack. A little later in life, we learn to walk. And this changes an overlapping set of neurons and synapses. And these guys have to change because these guys have changed. So then they still need to produce flexion withdrawal reflexes. And still later in life, one might acquire discus throwing. So a difference, an overlapping set of properties changes as well. And this one is really interesting because flexion withdrawal and walking are symmetrical. You can have the same changes on both sides, but discus throwing is very asymmetrical. So the prediction is, and this is something we're trying to test now, that there will be changes, asymmetric changes at all the levels, so that, and this will be a more complicated negotiation because there will be differences on the two sides, and therefore, as a result, flexion withdrawal reflexes will probably now be produced somewhat differently. They will still be good on both sides, but they'll probably be produced differently on the two sides, and similarly with walking. Again, walking will be intact, but it will be different on the two sides. And actually, we've just obtained the first little bit of evidence to support that. But in any case, the Heksors establish the central nervous system is in a state of negotiated equilibrium. So this is an example of how a new Heksor negotiates and changes an old Heksor. This is a rat walking along on a treadmill, and you see the right and left soleus EMG. When we make the reflex bigger or smaller, say on the right side, the soleus burst, the muscle activity is lower during walking, and it's bigger if we make the reflex bigger. But walking is not impaired. The animal still walks symmetric, is still balanced as it walks along. One hip is not lower than the other. EMG and kinematic changes maintain the symmetry in hip height. So here you see a smaller, if you make the reflex smaller, the ankle is not as plantar flexed. It's a little more flexed here. This angle is smaller, and therefore that alone would make the hip lower and the animal will be tilted. However, this angle gets bigger. As a result, the hip height is maintained. And conversely, and so that the kinematics are a little different, but the hip height, the key feature, is maintained. The animal is level as it walks along. Conversely, if this was all that happened with a bigger reflex, the hip would be higher on that side, but this gets smaller. So as a result, the hip height is maintained. The old locomotion Heksor adapts to the new H-reflex Heksor. As part of this negotiation, other H-reflexes change. Now, this is a rat in which we trained the H-reflex to be larger in the right soleus, and another one, we've made it smaller. You see the smaller H-reflex. If we look at another muscle, the right vastus lateralis, we did not intend to make any change in the vastus lateralis. The H-reflex here gets smaller, and the H-reflex here tends to get larger with down conditioning. This is presumably part of the associated changes that keep the hips, keep key features such as the hips intact. So, and this is obviously at a much higher level. There have been a lot of studies now, as you might imagine, MRI studies looking at what happens as somebody learns a language, how the substrate of that language develops. And a couple of those studies have looked at what happens to the original language when you learn a new language. So this compares the activation, the MRI activation that's found during articulation in people that have that speak two languages versus one language when they're speaking their original language. So these people are speaking their original language, and the area involved becomes larger for articulation and for retrieval. These are the areas of difference. And you can understand why that would be, because you have words that sound the same but mean different things in the two languages, and different words that mean the same thing in the two languages. There's going to be a lot of overlap and potential contradictions between the languages that have to be sorted out. So this is another example of the impact of really a new Heksor on an old Heksor. So this is basically the presentation of the basic idea. And what I want to do from here on out is to talk about some of the many implications and potential applications of this, with emphasis on one that I think would be particular interest to the group here. In any case, at this point, Marcus, are there questions that it would be good to answer now?
So far, I do not see any question in the chat, but just to let you know in the audience, if you have a question, use the raise hand function or ask the question in the chat. But I think it was quite clear. I also see the backup slides you sent to me where you added some further slides for explanation. So I think you made a really good point so far. Yeah. So if you have questions in the audience, use your raise hand or ask it in the chat, or you go ahead for now, Wolfgang, and we do it then towards the end.
Okay. So we go ahead?
Yes, go ahead.
Okay. All right. So this is the original paper. What's emphasized in this paper is the current evidence supporting the paradigm, and most important, the many ways to test its reach to other skills in people and animals. I mean, there's a lot of testing that's necessary. The paradigm obviously isn't, it needs a lot of investigation. And the point is that there are many ways to test its predictions, and we've started to do several of them. The paradigm offers new and potentially important answers to extant questions. The generation and function of spontaneous neuronal activity. I would guess that a lot of what we see as spontaneous neuronal activity is really Heksors in ongoing negotiation. And there are ways that could be tested. We'd like to do that, both in humans as well as in animals. The etiology of muscle synergies. These are particular combinations of muscles that typically participate in the same relation to each other in various kinds of activities. These are probably the result of Heksor negotiation. And again, that could be tested by demanding a new kind of combination of two muscles that normally fire together, and now creating a skill in which they have to fire in antagonistically. That presumably would have implications for other synergies. And the control of homeostatic plasticity. This is viewed as, oh, well, the nervous system doesn't want to undergo any change. Well, that doesn't really make a whole lot of sense, because the nervous system is always undergoing a lot of change. And this really looks like, more likely, homeostatic plasticity is the result of the Heksors maintaining their negotiated equilibrium and opposing changes in it. And again, there is already some evidence suggesting that. There are situations in which homeostatic plasticity would not be obtained, would not be maintained, because it would not be advantageous. Those kinds of experiments can be done. So this is a recent article that's come out now looking at the implications for neurorehabilitation. In this case, and viewed in terms of the new paradigm, the goal of neurorehabilitation is to enable damaged Heksors to repair themselves. Now, the standard strategy has been now for the past several decades, skill-specific practice, which can be effective. You practice walking, you practice speech, but this has two problems. One is widely recognized, the other isn't adequately recognized. The first problem, the capacity for plasticity, while there's a lot of it, even in the adult nervous system, is limited. No, cut axons aren't going to automatically regenerate in the spinal cord and establish connections, for example. So the new strategy is to augment generalized plasticity, to give increased plasticity, which the Heksor can then shape during skill-specific practice. The second and less recognized problem is that practice is not sufficiently specific, that all these skills are very complex, and there are many ways where plasticity that is available could produce them. But there's no guarantee, there's no little person in the head who decides exactly what plasticity occurs when and where to get the best possible result. So the new strategy that comes out of that is targeted plasticity, developing ways which can be production of a very simple new Heksor, like a therapeutic Heksor, say H-reflex conditioning, that will eliminate a roadblock and guide Heksor adaptations during skill-specific practice. So these are two new strategies that sort of fall out of the paradigm, and you wind up with a triple strategy therapeutic framework. Basically, most of this is all non-invasive, and it's already producing very striking results in terms of enhancing recovery of function. So this is for neurorehabilitation. This is the area that I think may be of particular interest to many in this group, looking at brain-computer interfaces to make brain-computer interfaces as reliable as muscles. At present, brain-computer interfaces lack the high reliability that's essential for many actions. Nobody who lacked a death wish would use a BCI to control his body or his wheelchair on the edge of the Grand Canyon. The BCIs don't have the reliability essential for an action like this. Neural engineering alone can't solve this problem. A BCI enables its user to acquire a new skill. Thus, research needs to focus also on neuroscience, BCI-based skills that need to emulate muscle-based skills. BCI reliability is an intractable problem. This is a study done not too long ago using increasingly sophisticated analysis methods to use from EEG, reconstruct the kinematics of an action. And clearly, with the better methods, you get better and better, higher and higher correlations with the actual kinematics. However, the variability between performances is still there. It's as high here as it is down here. The reliability is not any better. It's just about a higher level. There's still a great deal of variation. So how do we get rid of that? Now, as I've already defined, a Heksor is a distributed network of neurons and synapses that produces a muscle-based skill and changes as needed to maintain the key features of the skill. A BCI creates a synthetic Heksor. A synthetic Heksor is a distributed network of neurons and synapses combined with adaptive software. The network and software combine to produce a non-muscular skill, and they change as needed to maintain the key features of the skill. So these are the normal Heksors, as you saw this before, and this is a synthetic Heksor that's using neurons and synapses, and it's also using adaptive software. This is normal CNS function, a very crude diagram of normal CNS function, all the areas of the brain, and they all are continually adapting to provide appropriate control of muscles for all the Heksors that use this system. This is normal CNS function. This is the BCI system function. You have those same areas, and you have the BCI. These are always changing to produce appropriate function of the BCI to produce BCI skill, the BCI skills. So what do we have when we combine these two things? What do we have in the actual nervous system? So this is a synthetic Heksor. This is the combination. Now, when I first made this figure, I thought, I can't show this. This is ridiculous. But that's the point. It is ridiculous. This is the reality. These guys, you have the normal guys that are all adapting to control muscles, and they're all supposed to also be adapting to control the cortical output to properly control the BCI, so that the synthetic Heksor also functions. This is why they're not reliable. This is what's happening. So how do we fix this? There are a number of things that have been done already that can make the problem easier. Or less severe. A synthetic Heksor needs an appropriate starting point. Reliably, this is in monkeys controlling a cursor in 3 movements. This is from Don Taylor in Andy Schwartz's lab several decades ago. As a monkey gets better and with practice, the monkey starts out using the same cortical activity that controlled actual movement, is now controlling this cursor. And as the monkey continues to practice, the cursor control becomes better and better. These are two people who are paralyzed by high-level injury in Jose Millán's lab, getting better with practice on a very simple skill. The improvement in performance is shown by the increasingly dark color here over the central EEG electrodes. So they get better and better over months in practice. After starting with an appropriate point, an appropriate imaginary point, imagining the movement and then going forward. The synthetic Heksor needs better sensory feedback. Normally, BCIs to date have simply had visual feedback, but obviously a real movement, a muscle-based movement, gets much more complex, much more timely feedback. So you can do that by, say, in a robotic arm, converting sensor output to stimuli going into cortex, therefore providing much more detailed and timely knowledge about what's going on than vision. Synthetic Heksor also can benefit from signals from multiple areas. Normally, you use the simplest cortical neuron-based BCI uses cortical activity coming from motor cortex, which does represent the movement, but it would also be affected by shifts in attention and by unexpected somatosensory input. If you record in other areas, somatosensory areas, and prefrontal cortex as well, you can measure this attentional shift, the input of that, and also the effect of sensory input, and you can remove it, and you can get a more accurate image of the isolated motor command to improve performance. So that's another way that's going forward. So, however, there's still the problem of negotiation. A synthetic Heksor shares neurons and synapses with natural Heksors. Thus, it's affected by the ongoing negotiation that maintains a negotiated equilibrium. Because there's only one synthetic Heksor and it differs greatly from natural Heksors, the equilibrium serves the natural Heksors. The synthetic Heksor loses out. It's at the mercy of ongoing CNS plasticity that it cannot control and about which it has no information. Thus, its performance is unreliable. Now, this is a problem that's going to remain difficult and present for a while because we understand very little about the process of negotiation. It's presumably going on continually. How it works and how it could actually be joined by a synthetic Heksor. Without that knowledge, there's limited there's limited there's there are limited measures we could take to actually try to improve this integration, to allow the synthetic hexer to participate effectively in this negotiation. But there is one way that we're now starting to undertake, which is to try to compel the a muscle-based action and a BCI-based action to work together. You could either do this in parallel, in which you had a cursor that's being controlled in two dimensions, and one dimension is controlled by the BCI and another dimension by the muscle. You could also do this in series, where you have to go from here to here with the BCI, and then you switch to the muscle, and then the BCI, and you go back and forth to basically compel them to really introduce them to each other so that they would learn to work together. This presumably would help introduce a synthetic hexer into the negotiation. Hopefully, we will eventually know more about the actual negotiation and be able to do this more effectively. But this is something that could have some impact on the unreliability of BCIs at present. So in summary, the central nervous system substrate of a skill behavior is a distributed network of neurons and synapses we're calling a Heksor. It maintains the key features of its skill, which are the key features are a memory. Individually, Heksor plasticity ensures memory stability. Collectively, Heksor plasticity ensures a satisfactory negotiated equilibrium. I've talked a little bit about the potential reach of the Heksor negotiated equilibrium paradigm to other behaviors, to spontaneous activity, to muscle synergies, to homeostatic plasticity, to rehabilitation. There's also a concept of pathological Heksors. Are addictions, for example, or compulsions, pathological Heksors that could be addressed in the same way, using the same sorts of means we're using to help damaged Heksors get better, the idea of synthetic Heksors and BCIs, and we're also would like to think about how we would do this, look at Heksors in an adaptive neural network design. For example, have a network in which we were able to insert Heksors and see how they interacted. These issues can be addressed in human and animal studies, and whatever the fate of the Heksor negotiated equilibrium paradigm, the problem it tries to address affects every aspect of neuroscience, that skills are sharing neurons and synapses. So there have been many people at our center and National Center that have been participating in this. I've talked about many of them. Our support from NIH, from the Veterans Administration, from New York State, from the VA Medical Center, and from other private foundations. Thank you very much. If you have questions or comments, I look forward.
Thank you so much. That was really amazing, very, very interesting. I'm not such an expert in this field. We do some BCI research, but I fully understand what you do is mainly ECOG, or is it like immersive? When you use BCI technology.
Well, what I've done is mainly with EEG, but we have people in our group who are using ECOG and stereo EEG. So it's interesting what's happening with the units. People were initially very concerned with identifying individual units, and now basically they're using units more in a group fashion and going back to zero crossing. So it's sort of moving away from individual units. But anyway, but my involvement has mainly been in EEG.
Oh, that's very good. So you can run it also over longer time spans. There are many questions already in the chat. How should we go? Jake or whoever has a question, I recommend you, if you can, just share also your video. You can ask the question by yourself. We have a open setting. I see Jake is already unmuted, and then I will spotlight you, and then you can ask the question by yourself. Go ahead, Jake.
Sure. So my work is in proprioception, looking at muscle spindles and tendon organs and all that fun stuff. So one of my questions was you were talking about the adaptability of reflexes, and the ability to condition them based on task demands and things like that. So I was wondering in this Heksor-based framework, something like the gamma or the beta motor system that can modulate the sensory feedback that you're getting from muscle spindles. Would you look at that as a way that the CNS has to kind of condition those hexers in real time? Um, or would you look at that as part of the hexer itself?
Oh, okay. Yeah, I hadn't thought of that. I mean, normally, one of the reasons I didn't say for going to the H reflex was that we were getting the muscle, getting the spindle out of the loop, and we didn't have to worry about are we looking at the spindle. But I don't know. It's something clearly if it's part of. I mean, you're looking at the gamma motor neuron. The gamma motor neuron presumably would be part, if the gamma motor neuron is adjusted a certain way. Um, I mean, as a neurologist, I think of the blood-brain barrier as sort of a sacred entity. So that if you're within the blood-brain barrier, that you're part of the CNS. This would be in the CNS, and it would then be part of it would be part of the Heksor, and it might change. You might make the gamma motor neuron more or less sensitive. You might be adjusting it. Yeah, that would be would become part of what's happening and part of the thing that could change to adapt. Yes.
Mhm. Okay, thank you.
Yeah, we have the next question from Clara. Please go ahead. Okay, I do not hear
Hey,
you, Clara.
sorry, can you hear me?
Yes, please go ahead. Yeah.
Yes. Okay. Yeah, sorry. I think the question is maybe a bit far off topic, but I was wondering because I'm working with patients with movement disorders, if this would change anything, how we think about therapy or learning new skills in those patients.
Yes. Yeah, that's really, I mean, yeah, that's really a lecture on its own. It, yeah, people think of it's really interesting. People think of, the chronic nervous system as as something that's stable, or the damaged nervous system is stable. But it's going to be a very contentious environment, really more active than the normal nervous system, because normal nervous system presumably is functioning. You have a nice, smooth, negotiated equilibrium. Everybody's happy, etc. But here you have all these forces. You have damaged hexers. You have primitive hexers that have been released. You have diaschesic processes. Um, you have um, rehabilitation related skills such as driving a wheelchair or using an eye movement system or even a BCI. All these things are interacting. They're basically it's a very contentious environment. And our job in this is to help the damaged hexer to survive and thrive and function in that environment. And there are very definite studies showing that you get into competition. Dave Reichensmeyer has done studies showing that that as control gets regained, part of that control involves suppressing primitive hexers that have been released. Flexion hexers that were there primitively and got subsumed with the development of the full, corticospinal function. When that gets damaged, then the primitive ones come out. There's also really interesting work from Murata going back 15 years showing the competition that occurs and how we can, by practice and by other means, we can strengthen basically the good guys, the damaged hexer, so that it can recover. And their studies show very clearly situations where it does recover and others where there's less practice that takes place in which it doesn't recover. So this very much applies, and I mean all that's addressed some in our neuro rehab paper that's just come out. But yeah, it's really very, I could go on for quite a while about the damaged nervous system. Yes.
Thank you. Ralph, you also have a question, right?
Yes,
please go ahead.
John, thank you for your usual thoughtful. I mean, I've been the way you present this and the clinical implications. And when you start in the periphery, I can begin to understand as an old-fashioned neurobiologist. But then when you talk about Heksors in the CNS, I'm just saying that's orders of magnitude more complexity, and those synapses and those negotiations are suddenly tied into much more complexity. And so whatever the process is, do you think it's the same process in the periphery as the CNS, or you think it's going to have to be a different sort of understanding when you start crossing those boundaries? And of course, Heksors are going to have to do both. They have to cross boundaries anyway.
Well, if you're talking about the periphery as the spinal cord or outside.
Yes, the spinal cord and
the
muscles.
Yeah, the spinal cord is, well, the answer is yes and no, because basically there are situations, and we're running into this now, that, I mean, in the largest sense, the spinal cord and the brain are together and they're acting. I mean, the spinal cord is an advantage because we can kind of isolate it either both transiently as well as permanently in experiments and look at what's happening there. But it is working together with what's happening in the brain. So the Heksors really extend down into the spinal cord. On the other hand, as you're talking about, they get differently distributed during age, for example. In younger people, locomotion is basically functions at a lower level. As people get older, if they're successful older people, there's more corticalization. With the loss of musculoskeletal functions, things get done at higher levels as well so that they can continue to walk, but their Heksors are now basically more complicated or at least more distributed than they were earlier on. So there's really continuous flux, but looking within the central nervous system, the overall answer is that this is a unitary thing. And the reason we study it so much in the spinal cord is that it's easy to see there. And as I say in the paper, it's easy to define things. What is a Heksor? I mean, I can define a simple Heksor based on its key features. That's what totally defines it. So locomotion isn't a right leg Heksor and a left leg Heksor, because essential part of it is the coordination between the two. So it's one Heksor, but obviously it'll become much murkier as you go to higher levels and higher level things. Yeah, but I think that probably addresses it. But there is this within the central nervous system. It's more or less they're very much together. So
thanks.
Yeah, then we go ahead with Nadia, please.
Hi. Thanks for the amazing talk. I'd like to ask, is there any relationship between, do you think that the mirror-neuron system may modulate the plasticity of Heksors? So action of the mirror-neuron system, I mean, action observation, do you think that action observation can modulate the plasticity of Heksors and play a relevant role in the in this procedure of reshape, basically?
Yeah, I mean, the whole idea of imagery is, I mean, I don't know all of that literature, but it has potentially great use in rehabilitation. I mean, I know it's used athletically, but it has potentially great use. And one of the ways in which that can happen is we would like to think that EEG can be used to both detect what's happening with imagery and also to encourage it. This would be the use of BCIs in rehab to basically encourage the development of the correct kinds of EEG activity associated with, say, during walking. Can we associate, the swing, the various stages of walking? And that's something that we're now hoping to be able to do. We talked some to Kuntai Guan's lab about this, to basically for therapy of people who aren't able to walk, at least to begin with. So, yes.
Thanks.
Great. Charlie has a question. You want to go ahead?
Yes, Jonathan. Many motor system disorders are treated by off-label prescriptions. How does the Heksor figure into this? How would you modify a Heksor by external medication?
Oh, by external medication. Well, I guess you would think about, I mean, it could be medication that would basically affect behavior so that it might, for example, make something more difficult or easier to do or strengthen some part of a behavior as opposed to another, that would therefore motivate responses by this in regard to the Heksor. And the other is in terms of affecting specific locations, such as, the response to stretch, a very, very specific synaptic connection, so that you could then say, okay, we made this stronger, we made this weaker. It would basically be using drugs. I guess functionally, it would be similar to what's done with, for example, in treadmill training, applying treadmills with split belt treadmills with different speeds on the two sides to someone with a stroke, to basically emphasize changes that when you go back to the regular walking, improve function. But you could envision various ways of using a drug, I think.
Well, I was asking the question from a different slant. A lot of these drugs are prescribed. Oh, this helps other people, but they don't seem to know why. And I was wondering if the Heksor knowledge might help that.
Oh, yes, yeah, certainly. Yeah, I mean, I hadn't thought of that before. I mean, you might get insights just on the effects of the drug in different kinds of situations. Yeah, yes.
Thank you.
Thank you.
There was also a question by Mariam. Thanks so much, Charlie. But by Mariam, she was asking, when you do neurohabilitation, does Heksor suggest that it must target the entire distributed network, integrating like brain, spinal reflexes, and peripheral muscles, or does it like purely localize on brain injuries?
Does therapy or is
It's about the neurorehabilitation, so the therapy,
okay?
yes.
Yeah, there are basically, if you talk about doing multiple strategies, you would really combine, for example, when I talked about like a therapeutic Heksor, like H reflex conditioning could be used as a therapy. We've used it certainly in rats. In rats, when they have a spinal cord injury on one side, they get weak on that side. You can make it stronger by up conditioning the H reflex on that side. And that is really targeted on making this connection stronger and getting it to substitute for basically what's missing. And in humans, for example, in which there's spasticity, you want to make the reflex smaller. And therefore, for example, if you have someone who cannot engage in skill-specific practice because every time they put their foot down, they have clonus or they have foot drop. So they can't practice. If you can reduce that spasticity by down training the H reflex, you can then enable them to step, and then they can practice, and then many wider changes occur. And you can see that by making changes, and Iko Thompson has shown this in a variety of different ways, that you can see much wider changes occur. So we make this slight little change, which makes one thing better, but that isn't the most important thing that happens. That just triggers much wider changes. And you see changes in both legs, in the muscles of both legs, and walking in general gets better. But so rehabilitation becomes a combination of skill-specific practice and very specific targeting of various kinds.
Thank you. There are two more questions. Do you have time to take them, or how is your schedule?
Yes, that's okay.
Yes. So then Jessica, go ahead, and then Nomi.
Great. Thank you so much for the talk. It's making me want to revisit Gina Turgiano's work with bringing up homeostatic plasticity. But my question is, it's a kind of a nice spin-off on your last answer. You and Iko's work with H reflexes, but what are your thoughts on corticomuscular coherence for also maybe probing Heksor activity and function as a way to non-invasively begin to kind of evaluate?
Yeah. No, that's really something. Yeah, that's really important. It's something I've been interested in for a long time. We've never really gotten around to looking at it seriously. Did some of it with Dennis McFarlane years ago, but yeah, that's a big thing. And that's something you could think about conditioning in terms of making that better, or increasing certain coherence and and maybe de-emphasizing coherence that's not productive. Yeah, that's a very potentially fertile area.
Great, thank you.
Yeah.
Naomi, please go ahead.
Hi, I think you mean this, Naomi. So it wasn't really a question, Jonathan. I was just thinking that maybe with regards to Charlie's question, medication could kind of act on the metabolic components, thinking of it as a holistic, a Heksor as being the whole, everything that can influence the behavior, including metabolic components. That was all, maybe.
Yeah, yes. Yeah, I think both thinking of drugs, both in general like that, and also in terms of very specific effects. I mean, that's something that may have, may have effects, particularly in terms of the cognitive behavioral parts of this, that, I talk about applying them for sensory motor deficits, but this is work that Peter Bruner and John Willey of Washington University, who are part of our center, are engaged in looking at cognitive behavioral disorders such as PTSD. And if you were addressing, they're very much into looking at amygdala function and how you can modify that, and how you might do that, again, potentially, eventually, non-invasively, to improve recovery. Again, you could think of drugs, particularly maybe in that setting. So, yes.
And on that one as well, with PTSD, say if you're using neurofeedback and you're aiming basically to reestablish a homeostasis across the networks that are, the main networks like the default mode, etc., and the like salient networks. And then that's a bit like that negotiated equilibrium that Liz was saying was probably more like a steady state, or creating that homeostasis that's required for optimal function.
Right. Yeah.
I will. Thank you very much, John. That was a fantastic talk. Thanks.
Thank you, John. So, do you want to give a last take-home message for everyone? I saw originally you were hoping for something. Maybe you want to share your thoughts.
Yeah. Yeah, I mean, I just wanted, I mean, every time I give a talk, and it was clear on this one also that people, I learned something and stuff comes up that I hadn't thought about. So I mean that's really good. And I think this is really, I mean, the whole issue of that's brought up, there's just so many things about this that are unknown. I mean, there are a lot of things that need to be true that for this all to work, negotiation needs to be going on all the time. So therefore, the Heksors need to be getting feedback even though they're not active. So how do they do that? How do they use it? How do they change themselves even though they're not getting results? You don't ride a bike for 50 years, but you still can do it reasonably well. Clearly, it's getting feedback, it's updating itself. How does it do that without getting feedback? There are also all sorts of there are all sorts of therapeutic applications. I mean, the fact that we've studied both for disease in impaired situations as opposed to also normal situations has given a lot of insight, and a lot of this has come out of that. And the effect in rats, that, for example, that you can look at things that you couldn't look at in people. I think people really should think about, even probably neural network people as well, that to think about what happens in the disordered nervous system. We're now doing just begun really interesting studies, Jody Brangaccio in our lab, looking at phantom limb pain, which is something that people with amputations have. And we're finding that targeted plasticity looks like it can address that very effectively. That training cutaneous reflexes can address that very effectively. But I guess one thing that I'd say to people is they really should think about, if you want to understand the abnormal nervous system and the normal nervous system, there really is an advantage in looking at the damaged nervous system as well and thinking about it and comparing it. So, I mean, that would be something that really should receive more attention even among basic scientists than it does.
Thank you so much. I think with these words and also with your presentation, you have definitely sparked more interest among many of the people who listened. Also, I'm more and more interested to jump on parts of what you have said. So thank you so much for your presentation today. And I'll quickly conclude the webinar. Just to mention, there will be upcoming webinars. They're not yet announced, but if you follow INNS.org, you will see what's coming next. And there will be also other opportunities to interact with the society, the Neural Network Society itself. For example, there's still a chance for roughly a week open to nominate INNS awards. And it's typically awarded to great scientists in the area of neural networks in general. And that could be, as we had today, more about the neuroscience aspects, but it could be also computer science or the direction of physics. It's really a multidisciplinary area here, and we have had very great awardees in the recent past. So if you have someone in mind, you just need to be a member of INNS and you can nominate anyone. Whoever you nominate does not need to be even part of INNS, but typically somehow they are part of the Society of Neural Networks. And then, of course, we are looking forward to have a nicer discussion while physically meeting. There is the upcoming International Neural Network Society gathering, which is the International Joint Conference on Neural Networks. And that's this time again co-located with two other conferences in the bigger IEEE World Congress on Computational Intelligence towards the end of June in Maastricht in the Netherlands. specifically for people from Europe. I will be there, but it's also rather easily to reach the Netherlands from anywhere in the world because they have a very big airport. So if you didn't do so yet, consider registering. There will be very great, not only presentations and poster sessions, but also I'm personally very interested in these plenary discussions and where some really great, very recent issues are being brought up and some world experts are discussing about these. So join us in these different occasions and see you again soon. Thank you so much for today.